In 1951, a young African-American mother of 5 named Henrietta Lacks visited the Johns Hopkins Hospital complaining of vaginal bleeding. Upon examination, Dr. Howard Jones, a renowned gynecologist at the time, discovered a large, malignant tumor on her cervix. Mrs. Lacks began undergoing radium treatments for her cancer, which was the best medical treatment available at the time for cervical cancer. A sample of her cancer cells, which were retrieved during a biopsy, was sent to Dr. George Gay's tissue lab. Dr. Gay was a prominent cancer and virus researcher who collected cells from all patients who came to the Johns Hopkins Hospital with cervical cancer. Each sample Dr. Gay had collected died quickly in the lab. However, what Dr. Gay would soon discover was that Mrs. Lacks' cells were different from any of the other cells he had ever seen. Where other cells would die, Mrs. Lacks' cells doubled every 20 to 24 hours. These amazing cells were nicknamed HeLa cells, and the fact that they were immortal had a massive effect on the progress of medicine. Now you might be asking, how exactly did HeLa cells help advance medicine? The answer is that scientists were studying human cells long before they started using HeLa cells. They, however, had a lot of trouble keeping the cell line they were studying alive. A cell line refers to all the generations of cells produced from a specific culture of cells. Working with a single cell line was important, since it allowed researchers to verify their results and build on previous research. But when the cells in a line died, they had to start over with a new cell line. However, since Lacks' cells were different, they provided researchers with the first immortal human cell line ever grown in a laboratory. HeLa cells have helped scientists understand a variety of viral infections. Researchers infect cells with a virus like measles or mumps and observe how it affects them. Researchers were able to develop vaccines for infections such as polio and human papillomavirus using HeLa cells in their research. HeLa cells have also contributed to in vitro fertilization and chemotherapy research, and are used to study the effects of drugs, toxins, viruses and hormones on the growth of cancer cells without experimenting on humans. They have also played a crucial role in the development of the COVID-19 vaccines. As soon as scientists recognized HeLa cells' potential, they went into mass production and were produced in the world's first cell factory. Now, you might be wondering, what makes HeLa cells immortal? Well, HeLa cells have a massive number of mutations. One of those mutations causes an abnormal expression of the human telomerase reverse transcriptase gene. Telomerase reverse transcriptase is a catalytic subunit of the enzyme telomerase. Telomerase is a ribonucleoprotein that adds repetitive nucleotide sequences to the end of telomeres. Telomeres are sections of DNA found at the ends of each of our chromosomes. Telomeres consist of the same sequence of bases repeated over and over. This sequence is usually repeated around 3,000 times. Telomeres help organize each of the 46 chromosomes found in the nucleus of the human cell. They protect the ends of the chromosomes by forming a cap. If the telomeres were not there, the chromosomes may end up sticking to each other. Telomeres allow the chromosome to be replicated properly during cell division. Every time a cell replicates, the chromosomes are shortened by about 25 to 200 bases per replication. However, because the ends are protected by telomeres, the only part of the chromosome that gets lost is the telomere. Without telomeres, valuable parts of the DNA would be lost every time a cell divides. This would eventually lead to the loss of entire genes. Telomerase lengthens telomeres in DNA strands, thereby allowing senescent cells that would otherwise undergo cell death to become potentially immortal, as is usually the case with cancer cells. So, basically, telomerase reverse transcriptase is responsible for catalyzing the addition of a nucleotide sequence to the end of a chromosome's telomere. This addition of repetitive DNA sequences prevents the degradation of the chromosomal ends after multiple rounds of replication. Therefore, an abnormal expression of the human telomerase reverse transcriptase gene enables cells to proliferate endlessly. Up to date, about 60,000 scientific publications have cited the use of HeLa cells. Although Mrs. Lax eventually passed away on October 4, 1951, her cells continue to impact the world, saving millions of lives each year, and helping advance medicine. Thank you so much for watching the video, and we hope you found it informative. Please consider subscribing to our channel if you enjoyed the video.